Welcome back, everybody, to podcast 1B, Chemical Bonding in Biochemistry, part of our BiochemSerye Learning on the Go series.
It's great to be here.
As a biochemistry professor, I've seen firsthand how students can sometimes get a little lost when we talk about how molecules interact.
Yeah, it's a lot to take in, isn't it?
It is. It's a lot to take in. And one of the things that can be really confusing is chemical bonding.
Chemical bonding, it's so important, though.
It really is. It's the glue that holds everything together.
I always like to tell my students to think of atoms as being like people. Sometimes they like to be alone, but often they want to connect with others.
That's a great analogy because just like with people, there are different types of bonds that atoms can form.
That's exactly right, and that's what we're going to be exploring today. The two main types of chemical bonds, ionic and covalent.
Sounds like a plan. So where do we start?
Well, I think we got to start with the basics. Why do atoms even bother to bond in the first place?
Yeah, what's the point?
Well, everything in the universe wants to be stable. Atoms are no different. They want to be in their lowest energy state possible.
So it's kind of like a ball rolling down a hill.
Yeah.
Eventually, it's going to come to a stop at the bottom.
Exactly. And that's where the valence shell comes in. You see this outermost shell of electrons, it plays a really important role in how an atom interacts with other atoms.
So the valence shell is where all the action happens.
It is. It's where the magic happens. So how do atoms get to this stable, low energy state?
Yeah, how do they do it?
Well, for most atoms, it's all about getting 8 electrons in their outer shell. We call this the octet rule.
Oh, the octet rule.
Yeah.
Like an octagon, 8 sides?
Exactly. 8 electrons. Think about it like having all of your bills paid.
Oh, I like that.
Yeah, you're not stressing. You're stable.
Yeah, no more debt collectors knocking on my door.
Exactly. So atoms, they can gain electrons, lose electrons, or share electrons to achieve this octet.
Okay, so that's how they form chemical bonds.
That's it. So let's talk about ionic bonds.
Okay, so this is where we get into the opposites attract scenario.
You got it. With ionic bonds, we have a complete transfer of electrons from one atom to another.
Okay, so one atom's giving up an electron and the other atom's taking it.
That's right. And this typically happens between a metal and a nonmetal.
Okay.
A classic example is sodium chloride table salt.
Oh, table salt. Hi.
Sodium's a metal with 1 electron in its outer shell, and chlorine is a nonmetal with 7.
So chlorine is just 1 electron away from having a full outer shell.
Exactly. And chlorine, being much more electronegative than sodium, wants that electron really badly.
Electronegativity. Can you explain that?
Sure. Electronegativity is an atom's ability to attract electrons.
Okay, so chlorine is basically electron greedy.
Pretty much.
Yeah.
It wins the tug of war for that electron.
So sodium loses its electron and becomes positively charged, and chlorine gains an electron and becomes negatively charged.
You got it. And what happens when you have opposite charges?
They attract.
Right. That attraction, that's the ionic bond.
So they come together like magnets.
Exactly. And what's really cool about ionic compounds is that they form these three-dimensional structures called crystal lattices.
Crystal lattices, I've heard of those. Aren't those what make up gemstones?
They are. You see those beautiful repeating patterns?
Yeah, they're so intricate.
That's all thanks to the ionic bonds.
Okay, so ionic bonds are all about give and take.
They are. So now let's talk about covalent bonds, where atoms share electrons.
Okay, so covalent bonds are more like a partnership,
Exactly. They're sharing their electrons to achieve that stable octet.
like study buddies sharing notes.
Exactly. So there are 2 types of covalent bonds, nonpolar and polar.
Okay, what's the difference?
With nonpolar covalent bonds, the electrons are shared equally.
So everyone's happy.
Everyone's happy. A good example of this is hydrogen gas, H2.
H2, 2 hydrogen atoms.
Yep. Each hydrogen atom has 1 electron, they share their electrons, and now they both have 2.
Okay, so it's a win-win.
It is. But with polar covalent bonds, the electrons are not shared equally,
Okay, so why is that?
because one atom has a higher electronegativity than the other.
So it's hogging the electrons.
Yeah, kind of like in a group project where one person does all the work.
Been there.
So this unequal sharing creates partial charges within the molecule.
Partial charges. Okay,
Yeah.
I follow you. So what's an example of a polar covalent bond?
Water H2O is a great example. Oxygen is more electronegative than hydrogen, so it pulls the shared electrons closer to itself.
Okay, so oxygen gets a partial negative charge and hydrogen gets a partial positive charge.
Right, and it's this polarity of water that gives it so many of its unique properties.
Wow, so it all comes back to chemical bonding.
It really does. And one last thing before we move on. We need to talk about bonding capacity.
Bonding capacity. What's that?
It's the number of bonds an atom can form.
Oh,
So
okay.
hydrogen can only form one bond.
Oh.
Oxygen can form two, nitrogen three, and carbon can form four.
Carbon, the superstar of organic chemistry.
That's right. Carbon is the backbone of all those complex molecules that make up living things.
So it's all connected atoms bond to form molecules. And those molecules make up cells and tissues and organs and eventually whole organisms.
And it all starts with those fundamental principles of chemical bonding.
Amazing. So we've talked about ionic bonds, covalent bonds, and bonding capacity.
We have. And I think we've laid a good foundation for understanding how atoms interact to form the molecules of life.
I agree. So what's next?
Well, next we're going to dive deeper into the world of biochemistry and see how these different types of bonds play out in the context of biomolecules like proteins and DNA.
Sounds good. I'm ready for more.
Great. Let's take a short break and then we'll be right back. Welcome back to Podcast 1B, everybody.
So in the last segment, we talked about ionic and covalent bonds.
You did.
But, in reality, it's not always so clear-cut.
You're right. It's more like a spectrum. Some
Okay.
bonds have characteristics of both.
And then we have metallic bonding, too.
Right, metallic bonding, that's a whole other story.
Yeah, it is. It's like a whole family of bonds out there.
Each with its own unique properties.
I like that analogy. And it's these properties that determine how atoms interact with each other and form the molecules that make up our world.
Exactly. But it's important to remember that chemical bonds aren't static. They're constantly breaking and reforming in response to their environment.
So they're dynamic.
Very dynamic. It's like a dance.
A dance.
Yeah, the atoms are constantly moving and interacting with each other.
And this dynamic interplay is what drives so many biological processes.
It is. It's essential for life.
Yeah.
So let's shift gears a little bit and talk about how these different types of bonds play out in the context of biochemistry.
Okay, let's do it. Where do we start?
Well, how about proteins?
Proteins, those are the workhorses of our cells.
They are. They do so much.
And their structure is so important for their function.
Absolutely. And, that structure is determined by the sequence of amino acids and the interactions between them.
It's like a carefully choreographed dance.
It is. So amino acids are linked together by peptide bonds.
Peptide bonds. Those are a type of covalent bond, right?
Yeah. That's right. They form between the carboxyl group of one amino acid and the amino group of another.
And these peptide bonds are strong.
Very strong. They create the backbone of the protein.
So what about the other types of bonds?
Well, they play a role too. They help to shape the protein's three-dimensional structure.
Okay, so give me an example.
Well, hydrogen bonds can form between different parts of the amino acid chain.
Causing it to fold into specific patterns.
Exactly, like alpha helices and beta sheets.
And ionic bonds can attract oppositely charged amino acids.
Right. And then we have van der Waals forces.
Van der Waals forces, those are the weaker attractions, right?
They are, but they're still important. They help to stabilize the protein structure.
Wow, so it's like a symphony of interactions.
It is. And it's not just proteins either.
What else?
Well, DNA, for example.
DNA, the molecule of heredity.
That's the one. And you know that iconic double helix structure?
Yeah, it's so beautiful.
Well, that's all thanks to hydrogen bonds.
Hydrogen bonds holding the two strands of DNA together.
Exactly. And those hydrogen bonds are weak enough to allow the strands to separate during replication, but strong enough to keep them together under normal conditions.
So it's a perfect balance.
It is. It's amazing.
Right.
And within each DNA strand, the sugar phosphate backbone is held together by covalent bonds.
Right, so we see this interplay of strong and weak bonds in DNA, just like we do in proteins.
Exactly. And it's the same for all biomolecules.
So chemical bonds are essential for life.
They are. They're the foundation of everything.
Wow. So what does all this mean for our students?
Well, I think it's important for them to understand that this knowledge isn't just theoretical.
Right, it has real-world applications.
Absolutely.
Hmm.
Think about drug development.
How so?
Well, drugs interact with their target molecules through chemical bonds.
So if we understand how those bonds work, we can design better drugs.
Exactly. And diagnostics, too. Many diagnostic tests rely on the specific interactions between antibodies and antigens.
Which are governed by the principles of chemical bonding.
Right. It's all connected. And let's not forget about metallic bonding.
Metallic bonding. Yeah, we haven't talked about that much.
Well, it's particularly important for medical implants and prosthetics.
How so?
Well, metallic bonds involve the sharing of electrons across many atoms.
Right. That's what gives metals their unique properties.
Exactly, like their strength conductivity and malleability.
So these properties make metals ideal for use in medical implants.
They do. For example, titanium alloys are often used for hip replacements and other orthopedic implants.
Because they're strong and durable.
Right. And biocompatible, too.
Biocompatible. What does that mean?
It means that they can coexist harmoniously with living tissues.
Oh, okay.
Yeah.
So how does metallic bonding contribute to biocompatibility?
Well, it's complicated, but basically the way a metal interacts with the surrounding biological environment is influenced by its surface properties.
And those surface properties are affected by the nature of metallic bonding.
Exactly. So it's all connected.
Wow. It's
Yeah.
amazing to see how these fundamental concepts of chemical bonding have such profound implications for real-world applications.
It is. It's truly fascinating. And it highlights the importance of continuing to explore and unravel the mysteries of the molecular world.
Absolutely. There's still so much we don't know.
But that's what makes it so exciting.
I agree. So let's take a moment to reflect on what we've learned about chemical bonding and its relevance to biochemistry in medicine. Well, we've covered a lot of ground in this deep dive into chemical bonding.
We have.
We started with atoms, and now we're talking about the molecules of life.
It's amazing how it all comes together, isn't
It
it?
really is. It's like a giant puzzle.
And chemical bonds are the pieces that hold it all together.
Exactly. We've seen how those strong covalent bonds provide the framework for biomolecules.
Like the scaffolding of a building.
And then those weaker interactions like hydrogen bonds and ionic bonds, they add the details.
The finishing touches.
It's like building a house. You start with the foundation, and then you add the walls and the roof and the window.
Puzzle landscaping.
Exactly. And all of those different components work together to create a functional and beautiful structure.
Just like in biochemistry.
It all starts with those basic principles of chemical bonding.
And it's so rewarding to see our students grasp these concepts and apply them to real-world problems.
Absolutely. It's not just about memorizing facts
It's about understanding how things work at the molecular level.
and how that knowledge can be used to improve human health.
Exactly. Think about enzyme catalysis.
Enzymes. Those are the catalysts that speed up chemical reactions in our bodies.
And their activity depends on those precise interactions between their active sites and the molecules they act upon.
So even a small change in those interactions can have a big impact.
It can. It can lead to disease.
That's why understanding chemical bonding is so important for drug development.
We need to know how drugs interact with their target molecules.
So we can design drugs that are more effective and have fewer side effects.
Exactly. And this knowledge is also important for diagnostics.
Many diagnostic tests rely on the specific interactions between antibodies and antigens.
Which are, of course, governed by the principles of chemical bonding.
It's all connected.
It really is.
So what's next for research in chemical bonding?
Well, there's still so much we don't know.
What are some of the big questions that scientists are trying to answer?
Well, one of the big challenges is understanding how environmental factors influence bond strength and stability.
Like temperature and pH.
Exactly.
Yeah.
Those factors can have a big impact on how molecules interact.
And if we can understand how those interactions change under different conditions,
Mhm.
we can develop better drugs and therapies.
Absolutely. And another area of active research is the development of new materials.
Like biocompatible implants.
Exactly. We're always looking for new materials that are stronger, lighter, and more biocompatible.
And it all comes back to chemical bonding.
It does. The properties of a material are determined by the way its atoms are bonded together.
So the possibilities are endless.
They really are. We're just scratching the surface of what's possible.
That's what makes science so exciting.
It's a never-ending journey of discovery.
Well, I think we've given our listeners a lot to think about today.
I hope so.
Chemical bonding is a fascinating topic,
It is. It's the foundation of everything.
and it's essential for understanding the world around us.
From the simplest atoms to the most complex biological systems.
So thank you for joining us on this deep dive into the world of chemical bonding.
It's been a pleasure.
We hope you've enjoyed it and that you've learned something new,
And we encourage you to keep exploring the wonders of chemistry.
because the more we learn about the molecular world, the more we realize how much there still is to discover.
Absolutely. The journey never ends.
That's right. So until next time, keep those atoms bonding.
And those molecules dancing.
We'll see you on our next deep dive into the fascinating world of biochemistry.